So while there's been some huge wins, like HER2 and breast cancer and precision oncology, there's a lot of work to be done. As a practicing oncologist, I frequently struggle with balancing the potential efficacy and the adverse effects of treatment for patients. A big reason for that is because biomarker development has largely plateaued in relation to the fast pace of drug development, really highlighting a need for predictive biomarkers of efficacy. So how is tumor biology evaluated today? So a pathologist looks at an H&E slide, renders an interpretation, and as you can see, it's largely descriptive. And so it's hard to make individualized treatment decisions for patients based off of these qualitative descriptions. There's also targeted sequencing of tumors, which can inform the choice of systemic therapy, but does so without evaluating for the tumor microenvironment. And so clinically, what this leads to is even in the most ideal biomarker drug pairs, there's still a lot of variability in clinical outcomes, and that variability is due to the tumor microenvironment. So to illustrate, commercially available tools only measure the tip of the iceberg when it comes to tumor biology. However, many studies have shown that the tumor microenvironment is critical to tumor biology and can actually, compared to DNA sequencing, better inform treatment selection and predict response. However, our ability to deliver precision oncology to our patients is limited in how we can assess and quantify the tumor microenvironment. And so I'm here to tell you that we figured out a way to quantify this. So the answer may be right in front of us. So here's an H&E slide. We all know that there's very rich biology in this image. Here are some examples: a tertiary lymphoid structure, which is a marker of immune activation. Here are some mitotic figures, which are suggestive of a very aggressive tumor. And then there are also a lot of things on the H&E slide, such as fibroblasts, that are probably biologically important, but we just don't know the therapeutic significance. And so what this highlights is a need to extract and aggregate information from the entire H&E slide, because it's not really feasible for a pathologist to look at the entire slide in high-power view. Hence, this is why it's a problem perfect for AI. So we've developed a pipeline in which, using a large data set of H&E whole slide images and bulk RNA sequencing, we've developed a tool that, using the H&E slide, can predict different facets of the tumor microenvironment. In addition, we are using large language models to extract other clinically relevant data to add to the model to improve predictive performance. So using H&E, our model has been able to predict 10 different aspects of the tumor microenvironment, including immune, tumor, and metabolic pathways, to reasonable accuracy. Importantly, this is a task that pathologists are unable to do right now in current clinical workflows. As you can see here, these aspects of the tumor microenvironment have been pretty strongly linked to therapeutic efficacy for a broad range of therapeutics. And practically speaking, the ability to quantify the tumor microenvironment will enable us to have a more precise understanding of tumor biology, which will then translate into a better treatment selection for patients. But it's not enough to be correct. We also have to recognize what exactly the model is learning to make the prediction. So here's one such example. This is an H&E slide that was predicted to have a high degree of T-cell cytotoxicity. We asked the model what exactly it was looking at in the slide to make this prediction of interest, and as you can see, it highlighted a bunch of lymphocytes, which are suggestive of high immune activity, and it paid less attention to areas of the slide that are irrelevant for the task. This has a lot of therapeutic implications, both in informing a choice of existing treatments, such as checkpoint inhibitors and radiation, but also potentially in identifying new indications. For example, maybe tumor-infiltrating lymphocyte therapy, given that we know that the lymphocytes in this tumor are effective and cytotoxic. So what we propose, in addition to the existing pipeline, is taking the H&E slide that's already created as part of standard of care and scanning this, and then letting our AI tool do the rest. This can be pretty easily integrated in existing workflows, as slide digitization is an active effort within hospitals. So imagine the new possibilities for precision oncology with our tool that captures a more complete picture of tumor biology. In addition to better treatment selection for patients, another possibility is to pair this tool with existing assays. For example, one attractive use case is in combining this with self-recirculating tumor DNA to improve predictive performance of tumor relapse, which represents a value proposition to doctors and hospitals. In addition, imagine how much more efficient we can be in clinical trials. By using our tool to better select for patients whose tumor biology is more likely to respond to a novel investigational drug, we could potentially raise the response rate in earlier phase trials, which might save costs down the road due to expedited drug approval. In addition, we could also identify new indications for existing drugs, which represents an attractive value proposition to pharma companies. Cases like this is the future of precision medicine, precision oncology, and our technology is only going to get better with more data and resources. And so we're excited to commercialize our AI-enabled tool that can quantify actionable insights of tumor biology. Our first step is to further develop and validate our tool in the breast cancer space. While I haven't presented this data today, we've started applying this tool to specific therapeutics and are looking for additional data sets. We also have data that there are common spatial features on an H&E across multiple tumor types that promote therapeutic response, suggesting that our technology can be used for essentially virtually any solid tumor. And based on what's out there, we think this could be quite profitable. And given that our tool is a novel way to understand therapeutic resistance, we're also looking to partner with other drug developers to use our tool to potentially improve the drug development process for precision oncology therapeutics. And so in conclusion, we're seeking partnerships and financial support to improve our model using additional data, develop a user interface, and eventually execute trials using this tool as a precision biomarker, which we here at MGH think that we are uniquely poised to do this given our long track record of drug development and trials. I'd like to thank everybody who's contributed to this, and most of all, my family.